Okay, guys, so we are going to talk about proteins now. We talked about the DNA, nucleic acids last time. Now we're going to talk proteins. Proteins are made of carbon, hydrogen, oxygen, nitrogen, and every once in a while some sulfur. They can also be called peptides or polypeptides, and they are a chain of amino acids linked together by peptide bonds. Amino acids, there are 20 of them that are commonly used in the earth, and pretty much every living thing comes from those 20 amino acids. All the proteins needed to sustain life come from those 20 amino acids. There's a few others that pop in and out, but most of it comes from those 20. So we all use virtually the same set of amino acids. An amino acid's basic setup is this. You have your amine group, NH3, and that's why it's called an amino acid. And then you have your carboxyl group, and then you have your R chain or your side chain, your R group. This is what makes it special. They always have the amine group and the carboxyl group. The R group is different per each type of amino acid, so that accounts for the variability within the life on Earth. Here is a chart that tells you a little bit about each of the 20. If you look, the black, you have your amine group and you have your carboxyl group. That's on all of them. It's the same black part on each and every one of them. The blue is the R group, the side chain, and that R group changes how they react. Everything in this orange box is going to be nonpolar and hydrophobic. It's not going to want to dissolve in water. Everything else, the green, the magenta, the light blue, all of those are going to be polar and they're going to dissolve in water. The two in the magenta, these two have their carboxyl groups at the end, and the ones in this light blue, they all have amine groups at the end. And so that's going to change the way each and every one of them react and how they work in your body. Okay, so quick test. Glucose molecules are to starch as blank are to proteins. Oils, fatty acids, amino acids, or waxes. Did you say amino acids? I bet you did. All right. Which component of amino acids varies between the 20 different kinds? The amine group, the carboxyl group, the hydroxyl group, or the R group? Guess your answer. And did you say R group? Right. The amine and carboxyl group are always there, and the hydroxyl group can be part of the R group. All right, this chart. What this chart shows you is the difference in amino acids for the cytochrome C protein in there. It's not labeled properly, but that's what it is. So when you're looking at a horse versus a horse, they're the same thing. So they have 0 changes in amino acids. A horse versus a donkey has 1 thing. A horse versus a chicken, 11, penguin, 13, snake, 21. That's how you read this chart. So, okay, so the DNA sequence of horses and donkeys are identical. Well, I just showed you that there was 1 amino acid that's different, so they are not identical. Penguins have an increased number of hydrophilic amino acids in their cytochrome C protein. This table doesn't say anything about hydrophilic versus hydrophobic. It doesn't tell you which ones they are. All it tells you is how many are different. So we don't know that that's the answer. This table doesn't tell us. The snakes and horses use entirely different amino acids in their cytochrome C structure. If you look at snakes and horses, they have 21 differences. However, there's 104 in the cytochrome C protein. So that is not a completely different. It's got a lot of difference, but not completely. The last one, the gene for cytochrome C protein is in snakes is most similar to the gene for the protein in penguins. So you look at snake versus penguin, and there are, we'll look here, 17 differences. It is closer than anything else for a snake. It's further away, chicken, donkey, and horse. So the correct answer is going to be D. Now, protein shape. Proteins start out with a basic shape, and then they fold and fold and fold and fold, and to give them a primary, secondary, tertiary, and quaternary structure. The primary structure is just the sequence of amino acids: alanine, leucine, serine, lysine. That is a sequence. It's just amino acid after amino acid. Whatever string they come in is going to be your primary structure. It's kind of like the letters of a word. After that, the amine groups and the carboxyl groups, the hydrogens in them want a hydrogen bond to hydrogen oxygens want a hydrogen bond together, and either they will curl and make this helix, or they will crimp and make a beta sheet, a pleated sheet. And that has nothing at all to do with the R groups. It's only about the amine groups and the carboxyl groups, and whether it kinks or it twists. So here's a picture. You can see the yellow is a twisted portion of this protein, and the green is a pleated portion of this protein. The third structure is the tertiary structure. The tertiary structure, so you have either a coil or a pleat, and then it folds on itself. And now you're looking at the hydrophilic and phobic regions of the R groups. So the hydrophobic R groups want to get together, and the hydrophilic groups want to get together, and they are going to group, so they're going to fold on themselves. So this is a cluster now. We started out a long line, then it crinked or circled, and then it folded on itself on top of that. That's the tertiary shape, and this does have something to do with the R groups, unlike the ones before. Now, denaturization is taking that protein and changing its shape, changing the nature of it, because when you change the shape of a protein, you change what it does. What you can never change is the primary structure. That primary structure is not going to change because you're not changing the letters of the words. So if you heat it up, if you change the pH, if you change the salinity, the salt level, you can denature these proteins. And what's going to happen is whatever the glob for the tertiary structure is going to unfold back to that pleated sheet or that alpha helix, and then as it cools, it's going to renature, but it's probably going to renature differently than it did the first time around. So, and this is why cooking changes things. We all know what an egg looks like when you crack it, and then when you cook it. These proteins in this egg have denatured and changed shape, so that changes the taste, that changes the smell, that changes the texture, that changes just about everything about the egg. And that's why cooking changes the food so much. It's denaturing the proteins in there. It's also the difference between a raw steak and a cooked steak or a well-done steak. How much denaturing has happened. If you do a well-done, a lot of denaturing has occurred all the way through. If you do medium rare, then the outside has changed shape, but the inside is a little closer to the original. The last shape is the quaternary shape. Now, the quaternary structure is going to take one polypeptide tertiary structure and bond with other polypeptide tertiary structures to get together. Not all proteins will do this, only large proteins. This picture is an example of hemoglobin, and you can see like the red is one protein together, and the aqua and the silver are another, and then this would be a third or fourth, and they come together and they work together, working on those hydrophilic, hydrophobic interactions between each tertiary structure of the other one. So, the tertiary structure of a protein refers to its A, size, B, presence of pleated sheets, C, overall 3D structure, or D, the number of R groups it contains. Get your answer? Did you say C? Okay. The blank structure of a protein consists of a chain of amino acids assembled in a specific order. Is that primary, secondary, tertiary, or quaternary? Did you say primary? Okay. Scientists alter the valine molecule to prevent hydrogen bonding atoms to the R group. This would not affect what structure of the protein, the secondary, tertiary, quaternary, or all structures? The secondary, A. And that's because, remember, the secondary structure does not have anything to do with the R structure, or R group. It's only about how the amine and the carboxyls bond together, hydrogen bond together. All right, so here's a little bit about structure. The primary structure is a bond between the amino acids, and it is just a single straight chain. The secondary structure is the hydrogen bonds between the amine and carboxyl groups, and that's either going to give you the helix or the pleated sheet. The tertiary is the hydrophilic and hydrophobic clusters of R groups, and so that's it folding on itself, and that's going to make disulfide bonds. And then the quaternary is an attraction between multiple peptide chains, and it is not present in all proteins, only the larger ones that group together. Now let's talk types of proteins. Proteins pretty much do everything in your entire life, literally everything. It's all about the proteins. And when it comes to this class, it's probably down to a protein. So the things they can do, they can be structural. They can give the cell cytoskeleton its structure. It can give your hair cell its structure. They can be contractile and make your muscles move. It can also help like the flagella of a sperm move. They can be storage for amino acids so the proteins can get rearranged to make a new protein. They can be for defense, like the antibodies. The antibodies will protect you from invaders. Also, the membrane proteins. The membrane proteins are proteins on the membrane of a cell. And as those membrane proteins can do, and notice they're listed on 3 of them, but for defense, it can say, oh no, you don't get to come into the cell, and keep them out, keeping it safe. Transport. So you have hemoglobin, which will transport oxygen throughout the body. And you also have the membrane proteins, which again will say, oh, I need you to come in, or I don't need you to come in, depending on what nutrient or whatever it's looking at. Signaling is the next one. Hormones are going to be a type of protein that signal your body to do things, like testosterone and estrogen signal puberty. And then there's also the membrane proteins that signal, hey, I need more water over here, or hey, I need some potassium over here. The last one is enzymatic, and enzymatic regulate the speed of chemical reactions. These are proteins that speed up a chemical reaction. So, hormones are an example of what class of proteins? Structural, defense, transport, or signaling? Did you say signaling? Good. Hemoglobin is an example of what class of proteins? Transport, signaling, enzymatic, or structural? Did you say transport? Good. All right, and that's where we're going to end today. So today we talked about proteins. We talked about how they are made up of amino acids. We talked about the structure of an amino acid. We talked about the structure of a protein, its primary, secondary, tertiary, and quaternary. And we talked about the different functions of a protein. Okay, that's it. Have a great day.